There are 3 accessory organs in digestion: the liver, gallbladder, and the pancreas. They're called accessory organs because food doesn't travel through them, but they assist in digestion. The liver produces bile, which is a brownish fluid, which is then stored in the gallbladder. Bile is a fluid produced by the liver to help emulsify or physically break down fats. It provides more surface area for lipase to work. So I have a large fat globule broken down by bile into smaller pieces of fat, and then lipase can break those down into glycerol and 3 fatty acids. The gallbladder stores bile, which is made by the liver, and secretes bile into the small intestine when a person eats fatty foods. The pancreas secretes pancreatic juices to aid in digestion. The pancreatic juices contain ALTA, which is an acronym for alkaline salts, which are basic, lipase, trypsin, and amylase. The alkaline salts go to the small intestine through the pancreatic duct, where they buffer or neutralize the HCl from the stomach. It raises pH in the small intestine to 8. Lipase is an enzyme digesting lipids into fatty acids and glycerol. Trypsin, which is a type of protease, is an enzyme secreted in inactive form, trypsinogen, which continues protein digestion. This is activated by the alkaline or basic pH. Pancreatic amylase is an enzyme which continues the digestion of starch into maltose units and completes the digestion that started of starch in the mouth. The liver is the largest organ in the body. It receives blood from 2 supplies: the hepatic artery, oxygen-rich blood from the heart, and hepatic portal vein, which is nutrient-rich blood from the intestinal walls. The liver acts as a gatekeeper to absorb nutrients from the food. The liver removes excess nutrients in the blood. It also makes plasma proteins. It manufactures important blood proteins, for example, fibrinogen for blood clotting. It has excretory functions as well. It breaks down old red blood cells. The pigment, or the color, of the red blood cells is excreted in bile. The iron from red blood cells is stored to make future red blood cells. The liver detoxifies. It breaks down poisons and non-food substances in the blood. For example, alcohol, caffeine, nicotine, drugs, and excess hormones. It also produces urea through the process of deamination, the breakdown of excess amino acids from proteins. The digestive function of the liver: it produces bile, which emulsifies or breaks down fats, which increases the surface area for lipase to act on. Bile is stored in the gallbladder. The liver also stores vitamins and minerals. The liver takes up and stores fat-soluble vitamins such as A, D, E, and K, and inorganic minerals such as iron. The liver removes excess glucose from the blood after meals and converts it and stores it as glycogen. Once glycogen stores are full, it converts extra glucose into fat, which is released from the liver into the blood and stored in adipose, which is fat tissue. When blood sugar levels fall, the liver converts glycogen back into glucose and releases it into the blood. The liver is stimulated by hormones from the pancreas. Insulin is released after a meal to cause conversion of glucose to glycogen in the liver for storage. After not eating for a while or fasting, glucagon, another hormone, is released from the pancreas to convert stored glycogen in the liver back into glucose. Summary of functions of the liver. It makes proteins like fibrinogen, has excretory functions like deamination, which makes urea, the main component in urine, detoxification, removal of drugs and alcohol, and breaks down old red blood cells. D is its digestive function. It makes bile. And S is for stores. It stores glucose as glycogen and vitamins and minerals.